Okay, good afternoon, everybody. Let's complete our first block of classes, looking briefly at the history of microbiology. And we've learned a lot of important things along with those old microbiologists because following the work of Pasteur, we've learned that microbes are everywhere. They are ubiquitous in the environment and they do many different things, as Pasteur was able to show. And Pasteur was also able to show that it was very, very easy for things to become contaminated with microbiome, with bacteria. Um, and the microbiota is able, once it contaminates something, is able to reproduce very, very rapidly. All cells come from other cells, and they come from other cells extremely fast, as we're going to learn when we talk about bacterial reproduction. And this can cause tremendous problems. But in actual fact, until right the way through the 19th century, people were still arguing about the ability of bacteria, for example, to cause disease. And although by the 19th century, bacteria had been visualized using compound microscopes, they'd been shown to be ubiquitous, they'd been shown to do all sorts of different things, still people doubted their ability to cause disease and result in human mortality. Now, in the 18th century, this gentleman, Ignaz Semmelweis, was one of the first to really demonstrate empirically that there was an association between an infectious agent and disease. He didn't understand completely the mechanisms, although he strongly suspected that bacteria were involved. Ignaz Semmelweis was an obstetrician. He was working in Vienna in obstetrical wards in the hospital in Vienna. And as in all European hospitals, or hospitals all over the world, hospital was one of the worst places for a woman to give birth, because there was in all of those hospitals a background of a disease called puerperal sepsis. Now, sepsis is a particular condition. It results from bacterial infection and is actually a form of shock. Now, that is shock. That is clinical shock. That is a catastrophic drop in blood pressure as a result of widespread infection throughout the body by bacteria. They didn't understand the mechanism of that at all. And it was observed, for example, that women gave birth in hospital were at a risk within 5 to 7 days of developing a rampant infection, which in many, many cases resulted in death. And just to show you, first of all, in all of the hospitals, this ran as a background. There was always this puerperal sepsis present. This blue line here is actually a hospital in Dublin, very remote from where Semmelweis was working. But the pink line here is the hospital where Semmelweis worked. And you can see the two, Dublin and Vienna, are very, very similar. But then the hospital in Vienna did something new and different. And that was they began to autopsy women who died of puerperal sepsis. And that began here. This line here shows you the date on which that procedure started to be used. And immediately that happened, immediately they started to autopsy women, the puerperal sepsis rates just exploded. Look at the pink line now and see what the mortality rate due to sepsis is. It's double, triple, quadruple once they began this procedure of autopsy. And the reason for that is very easy to understand, because surgeons in those days did not observe sterile procedure. They would move from one patient to another without washing their hands. They would move from examining a woman who just died of puerperal sepsis to one who was busy giving birth and thus infect her. And in addition, now they began to autopsy women, and dead bodies were essentially culture vessels of the bacteria which had caused the disease. The surgeons not only did not wash their hands, they wore filthy clothes, which literally transported bacteria into the wards and from patient to patient. And that is why the number of cases just exploded. Now, Semmelweis was very disturbed by this. He strongly suspected that these newfound bacteria were actually responsible, but nobody would believe him until something really dreadful happened. And that was that a friend of his who was working in the same hospital was autopsying a woman and pricked his finger. And within a very short time, he developed a rampant bacterial infection and died. And this man died of symptoms very similar to the women who died of puerperal sepsis. Now, what was happening was women were becoming infected during the process of childbirth with bacteria, in particular Staphylococcus or Streptococcus, which you'll hear about later. Those bacteria were growing, the populations were exploding inside their reproductive organs and then escaping via the fallopian tubes into the abdominal cavity and thence to the rest of the body. And as a result of that infection, the body has a particular immune response, which results in this catastrophic drop in blood pressure. And that is sepsis. Sepsis is by definition a catastrophic clinical shock due to the dropping of blood pressure, and then organ failure and everything else, and women very rapidly died as a result. Ignaz Semmelweis was so disturbed by the death of his friend that he immediately made that association. He made an association between that kind of infection and the symptoms that he saw. And he began to institute in his wards, anywhere that he was in charge, a regime of washing hands. He washed with a very simple disinfectant, which was simply chlorine dissolved in water, which is effectively bleach. And he insisted that everybody wash their hands between patients and then on entering the ward and on leaving the ward, and that they wear clean clothes. And the rates of puerperal sepsis plummeted in his wherever he was in charge. Unfortunately, the other doctors didn't like this idea at all. And Semmelweis was eventually fired. It was partly his own fault because he was actually a very unpleasant character, apparently. Very difficult to deal with. But he died a sad, lonely death eventually. But his work lived on. His work actually became well known after a while. And a number of people now began to act upon this kind of information. In particular, there was one surgeon in Britain who we'll hear more about, called Joseph Lister, who heard about Semmelweis's work and similar work and instituted sterile procedures in his operating room. And those massively lowered the rates of infection of patients that he operated on. So a lot of this kind of evidence, empirical evidence for an association between bacteria and disease began to be accumulated. But it needed scientific experimentation to actually demonstrate that association. A lot of people did not believe it. They didn't believe that these small things flying around in the air could cause disease. So that theory that bacteria or infectious agents can cause disease is called the germ theory of disease. Initially, it was associated purely with bacteria. They didn't know anything about viruses. Later on, we learned that there are other germs, other infectious agents, which can also cause disease, in particular viruses. For the moment, our attention will simply be on bacteria. So you've heard about Pasteur already. Pasteur demonstrated the ubiquity of microorganisms. He demonstrated the ability of microorganisms to cause contamination. And from that, we could infer that bacteria could be transferred from patient to patient or from wound to into wounds or whatever and cause disease. This man, Robert Koch, was as famous really as Louis Pasteur, and they were huge rivals. But Koch was a German. Pasteur, of course, was in France. And Koch was very interested in trying to establish an association between specific bacteria and specific disease. He believed that specific bacteria would cause specific symptoms. And he worked in particular on a very, very dangerous organism called anthrax. Now, anthrax is a bacillus. It's called Bacillus anthracus. And it causes the disease of anthrax. And anthrax is an extremely dangerous disease. It had two massive impacts on human populations. The first was it infected cattle and sheep. And so it killed livestock. But second of all, from those corpses of the livestock, human beings could be infected. And if they were infected, in one particular way, if they received the infection through, this is my cat. Fortunately, he doesn't have anthrax, but his tail's very irritating. Go away, please. So in one way, if you inhale anthrax and get a pulmonary infection, it's almost invariably fatal. There's another form of anthrax, which is on the skin, which is not as dangerous. Now, Koch was very interested in this because he could go to the cattle or sheep or whatever had died, and he found that the corpses were full of one particular kind of bacterium. It has a particular characteristic, and that is it produces spores, and the spores are very, very characteristic. You can identify anthrax cells quite easily. So he looked in these dead cattle or sheep or whatever, and he found these particular kinds of bacteria, and he made the association. But now he's got to prove it scientifically. And he set up a series of experimental steps to demonstrate, first of all, an association between anthrax bacilli, the actual cells, bacterial cells, and the symptoms of anthrax. So here's how he hold on, I'm just going to jump here. So he, first of all, he worked on sheep, and he extracted from sheep these bacilli. And he was able to grow them in culture. Now, this is very important. He was able to establish principles whereby he could develop pure culture. And a pure culture is one where you have a medium. A medium is something that you grow organisms in. And in that medium, he had only one kind of bacteria. That is a pure culture. So pure culture, usually we attempt to get them to develop from one cell, because a bacterial cell will divide and produce two identical bacterial cells. Remember Virchow and the development of cell theory. One cell always comes from another cell. Cells grow to a certain size and divide and produce two identical cells. Those two then divide for 4, 8, 32. Very, very soon in a culture, you will have a gazillion bacteria, which are all identical. They all have exactly the same genetic background. So this is what he did. He developed methods to produce a pure culture. He then took that pure culture and he injected it not into sheep, but into an experimental animal. He actually used white mice. And he injected into the mice, and he demonstrated that the mice died. And they died with very distinct symptoms. And those symptoms were the same symptoms that that sheep die of when they have anthrax. He's not finished yet. He's made a pure culture extracted from bacteria taken from a corpse. He has made pure culture, injected it into an experimental animal. The experimental animal dies of the same symptoms that the original death was caused by. And now his final step is to extract bacteria from this experimental animal and demonstrate that they are the same bacteria as he originally extracted here. Here's anthrax, it's Bacillus anthracus. Remember that name. And it has, you can see here in that space in the middle there, that is a spore. And it is carried in usually in a terminal position there, which is very typical for anthrax. So you can recognize the cells very easily. Extracts them from a dead animal, cultivates them in pure culture, infects an experimental animal, and from that experimental animal extracts the identical bacteria again that he found here originally. He's demonstrated that these particular bacteria cause particular symptoms, and that those bacteria, if you inject them into other animals, will cause the same symptoms. And you can then extract the same bacteria from that experimental animal. These are called Koch's postulates, and they remain very, very important in clinical medicine. If you want to, if you find a new infectious agent, and you want to demonstrate that that infectious agent is causing a particular disease, you usually have to press it through all of Koch's postulates in order for other scientists to agree that that is the infectious agent. It still applies. You must please understand this process of Koch's postulates. You will be asked about it, and it remains a very important feature of clinical medicine. So with all of this work going on, I got this a little bit out of order, so I'm just flipping through quickly. Okay, with all of this work being done on infectious agent, on the role of bacteria as infectious agents, people all over the world began to be interested because infection was the major cause of death in hospitals, and in particular where surgery was involved. First of all, most surgery was an extremely brutal operation. Surgeons employed very big, tough men to hold down their patients while the operation was going on, because, of course, apart from alcohol and maybe some opium, there was very little anesthetic. So people were operated on fully conscious. So the surgeon, in order to get the work done, had to work very, very quickly because the patient would be writhing and screaming, carrying on. Under those conditions, particularly the old surgeons who didn't wash their hands and everything, the rates of infection were absolutely enormous. And surgeons were really regarded as being the lowest of the low in the medical profession because of that. They had very, very, very high mortality rates. They were, in general, very filthy, rather tough, and hardened because they'd had to listen to patients screaming under their saws and knives for their whole careers. It was a very unpleasant procedure, having surgery, until two things happened. And that was in just past the middle of the 19th century. First of all, anesthetics were invented. And they discovered the initial anesthetic they used was ether. And people could be anesthetized with ether, rendered unconscious, so they felt no pain, no writhing, no screaming, no nothing. They stayed still. Infection was still a major problem, however. But this surgeon here, Joseph Lister, heard about Semmelweis's work, heard about Koch's work, paid attention to what Pasteur was saying, and decided that he was going to set up an operating area an operating room effectively with no bacteria or as few bacteria in it as possible. He operated in small rooms, which he filled with a fog of carbolic acid, phenol. A very, very, very unpleasant working environment and very dangerous for the clinical workers who operated in it. But he also, they didn't yet know about things like wearing masks. Here he is operating in his operating room. And they had a little, they called them atomizers. They had little things that sprayed a mist of carbolic acid. It's a very effective disinfectant. They covered their patients with cloths which had been soaked in carbolic acid. They washed the skin and everything with carbolic acid. So they really had spectacular results in reducing the rates of infection. And in doing so, he demonstrated to medicine, along with the work of Semmelweis, etc., he demonstrated that it is bacteria in the environment of the hospital or of the operating room which were causing the massive infections which had formerly been killing patients. They still lost patients from infection. It didn't disappear by any means, but the rates were spectacularly reduced. And these kind of methods very rapidly spread until surgeons actually acquired some respectability, and the respectability that they have now. However, I'll tell you that in old days in medicine, the respectable doctors were the ones, who didn't indulge in operations and everything, and they were called doctor. But the surgeons were so despised, they never acquired a title, and they were always called Mr. And you will still find that. You will still find surgeons who insist on being called Mr., even though they are highly respectable and they charge exorbitant fees. So, okay, so Koch established finally the very clear association between specific bacteria with specific symptoms, which were reproducible in experiment. So I must just explain to you, this pertains to the work that you're going to be doing in lab. They had a tremendous difficulty because there was an essential step in establishing Koch's postulates, which was to make a pure culture, to cultivate a pure culture. Now, Koch actually did it laboriously through liquid culture. It's very difficult to establish pure culture through liquid culture. But it's actually very, very easy to establish pure culture on solid medium. And solid medium is a nutrient broth of some sort with a solidifying agent in it. And the reason that it makes it so much easier, you will discover in lab. You can take a very small amount of bacteria, a sample, put it onto a solid surface and spread it. And what you're doing is you're diluting effectively. You're spreading bacteria away from them. Now you take a very small part of that, spread that, and you're spreading very few bacteria far from one another. Then you do it again, you spread again, and now you're taking a very, very small number of bacteria and spreading them very far apart, so far apart that you'll separate individual bacterial cells one from the other. And when you look at your solid medium, what you're going to see from that one cell, a colony of cells will grow. And you can pick that colony and be sure that what you're looking at are genetically identical cells. They have formed clones, which ultimately form a visible dot on the surface of the solid medium. But here's the problem. That's that solid medium. How do you solidify a nutrient broth? Well, when they first tried it, they used gelatin. Gelatin is a very good solidifying agent. That is what Jello is. But there is a problem when you use it in bacteriology, and that is that many, many, many animal pathogens love gelatin. It's protein. It's an animal protein, and they digest it. And very, very quickly, a solid medium, gelatin medium, can turn to mush, and all of your cells just mix up with one another, and you've lost your attempt to produce pure culture. But here's an early contribution from an American. And this is Fanny Hess. She worked in Robert Koch's, sorry, she didn't work in the lab. Her husband worked in Robert Koch's lab in Germany. And Fanny Hess was a good 19th century housewife. She made all her own jams and jellies and everything else. And she used a particular solidifying agent, which is called agar. Agar is an extract of seaweed, and it's commonly used in jelly making and jello making and everything, where what you're using doesn't contain a lot of its own proteins and everything. Normally, a jelly or a jam, you use the proteins from the jam, from the fruit itself, actually end up being boiled out and then solidifying. But if they're difficult to solidify, many housewives in those days added the substance of agar. Now, agar is extracted from seaweed. Most pathogens have never seen agar. They've never seen the substance that solidifies the liquid when you add agar. And as a result, she suggested to her husband, who was having great difficulty establishing pure cultures, she suggested to him that he use agar. And this is ideal. It's absolutely ideal, and that is still the standard. You will be using agar plates in lab, and that is the standard clinical microbiological practice. We solidify nutrient broth with agar, and we produce the solid plates, which are poured into shallow dishes called Petri dishes. And those you've probably seen already in lab. If not, you soon will. So thank you, Fanny Hess. Okay, so that set in motion all of the clinical microbiology, diagnostic microbiology, and also enabled clinicians to begin to work in relatively safe hospital and clinical environments, as we understood how to manage the principles of contamination and sterile work. But it did very little if people were liable to be infected. It did very little. There was no antibiotics or curative agents that you could give people if they became ill. But there was one instance already known where we could prevent illness. And this was in the case of smallpox. Now, I should explain to you that the smallpox is a viral infection, not a bacterial, but it's a viral infection. It is a dreadful, dreadful disease. And in former times, it was widespread all over the world, and it was absolutely dreaded as one of the great plagues of humankind for two reasons. First of all, it has a very high mortality rate. It is extremely infectious, and it produces these large pocks on the skin all over the body. And these pocks have the potential to be terribly, terribly disfiguring. So in ancient times, smallpox was absolutely dreaded. We know that in two civilizations, hundreds of years ago, a system was developed whereby human beings could be made resistant to the infectious agent. And these systems were developed first by Arab doctors, and then we know that Chinese doctors also completely independently had developed a way of doing it. So the way in which they did it was that they would go to somebody who was recovering from smallpox. This was very important. The person had to be well on their way to recovering from disease, but still have pox. And they would take liquid from these pocks. They would break them open, and they could actually even dry the serum that there was, or pus that was produced. And they would use that to deliberately infect people. They would scratch that material into the skin of an uninfected person. And what they hoped would happen, and what happened most of the time, was that those people would develop smallpox, but they would develop a very mild case of smallpox. The reason for that was towards the end of an infection, of an illness, of viral infection like smallpox, the virus is weakened. The person has survived. Their immune system has fought the virus so vigorously that they weaken the virus. That is called attenuation. It's still a principle in vaccination in certain instances. But in those hundreds of years ago, those doctors were taking a tremendous chance, and they had quite a high mortality rate. But the mortality rate after these inoculations was lower than the mortality rate of smallpox itself, of a smallpox epidemic. So people were often willing to take that risk. In particular, the system was made famous by the wife of a British diplomat who had been in Turkey during an epidemic of smallpox. And she had gone to a Turkish doctor and had herself and her children inoculated using this old method of just using pus, whatever, from somebody who had recovered from the disease. And all she and her children then returned to London, and there was an epidemic in London, and she and her children survived that epidemic without being infected at all. And they began to use that method, but it was soon overtaken by another one. And this was a completely serendipitous observation by this man, Edward Jenner. And Jenner learned that women who worked as milkmaids never got smallpox. First of all, that was like folk tale, that milkmaids never got smallpox. But he went and investigated, he found it was actually true. But what he also found was the cows have their own pox disease. It's a very mild disease, but it's transmissible to human beings. But all that happens is that the milkmaids would develop a rash on their hands where they'd been handling the udders. And the infectious agent was transferred from the cow to humans. The milkmaids didn't really get ill, but they never got smallpox. The reason for that is that the cowpox virus is very similar to smallpox. It's so similar that our immune systems recognize when they have seen cowpox, they develop immunity, the body develops immunity. And the antibodies that we produce to cowpox can neutralize the actual smallpox viruses if ever that person is exposed to smallpox. So Jenner began to do this deliberately. He began to extract the fluids, both from milkmaids and from the cows themselves, whatever source he could get. He would simply get serum from these small pocks, and he would dry it, and he would then inoculate people with that, rather than with live smallpox virus, which was the Arab Chinese method. So the word vaccination actually comes from this word, which is vacca, which means cow in Latin. And he called this process vaccination. At first, people were very scared to do it, but then they saw the spectacular success. People who were vaccinated had a very, very, very high resistance to smallpox, and it soon became the norm. We still use a somewhat similar method to produce a smallpox vaccine, But in fact, we no longer need to vaccinate against smallpox for the simple reason that smallpox no longer exists on the planet as a wild disease. There are a few labs which maintain smallpox vaccine and live smallpox virus, but it is under strictly controlled conditions, and the disease no longer exists in the wild because of the success of the vaccination against smallpox. The protection which is produced from vaccination is called immunity. And of course, this is now a principle, although there are many different, I would warn you that there are many different ways of vaccinating people, of developing vaccines. We'll talk a little bit later on in the course about some of them. But the protection that they offer is called immunity. And it's always the aim that you produce long-lasting immunity, so that if people are ever exposed to the disease that they have been vaccinated against, their immune systems immediately react and prevent the infection from becoming established. So that was a way of preventing disease. So now we're now getting an advance on pathogens, because first of all, we recognize where they're coming from. We recognize how to establish what particular bacteria cause particular diseases. We have ways to limit contamination, both and exposure, and also the methods of vaccination to try and prevent. Pasteur, by the way, went on to produce a whole range of vaccinations, which we will talk about. And vaccination began to be used for all sorts of, you can use principles of vaccination to vaccinate against many different viruses, but also against bacteria. But what was really wanted was a way to cure disease. Once a disease had become established, how could you actually deal with it and stop it from proceeding so that the person could heal before they were seriously injured or died? And this took an enormously long time. The very worst thing you could do for most of human history when you had an infection was go to a doctor, because they used methods which very often had far more damaging effects than the disease itself. So now, for example, they would use things that were effectively poisons. They very commonly used arsenic. They even used really, really potent plant poisons, all to try and kill what was causing the disease, but usually ended up killing the patient. They also had strange ideas, of course, I've mentioned before, that the body was controlled by humors. And so they would bleed patients, they would extract blood from them in order to try and balance their humors, or they would induce vomiting and diarrhea in order to balance the humors. All of these things could be extremely damaging if you were dealing with somebody who is infected with an infectious agent like a bacterium or a virus. But the search began for really effective means to combat disease. And that is that is the science of chemotherapy. And its advance led to what's called the second golden age. The first golden age of microbiology was all of that tremendous exploration by the scientists like Pasteur and Koch. The second golden age of microbiology was when we began to use chemotherapeutic agents to limit infection when it had already been established. And of course, for us, the most familiar chemotherapeutic agent are the antibiotics, but there was a long history to their development. First of all, the very, very, very first sort of effective chemical agent was actually a natural agent. It was quinine, which is extracted from tree bark. And that was, again, an empirical observation Indian tribes in South America used the bark of a particular tree to combat malaria. And it was found that that was actually an extremely effective way, not only of curing malaria, but of preventing it. If you take quinine as a prophylactic, you can prevent infection. Quinine, actually, but I should mention quinine is actually quite toxic in high doses, not in low doses, but in high doses. This man here is one of the most famous of the modern microbiologists. His name is Paul Ehrlich. And Paul Ehrlich fantasized about finding a compound which would kill all infectious agents. Paul Ehrlich, by the way, knew nothing about viruses. He only dealt with, he thought he was always dealing with bacteria. And his magic bullet would kill pathogens, but not affect the host at all. And he was the first person to develop a synthetic drug. And it actually was developed from arsenic. Arsenic, as I told you, has been widely used for many years as to try to treat people, but it almost invariably killed people. In fact, it probably killed Mozart, for example. But Ehrlich developed this drug. It's called salvarsan to treat the disease of syphilis. Up until that time, syphilis entered the human population in Europe around about 1500 and had been a plague ever since. And he developed this drug which effectively treated syphilis, although it itself was quite toxic. Its toxicity could be managed and it could be used to cure syphilis. It's the first time that a synthetic drug had been developed which would combat an infection. In the 1930s, the first truly effective antibacterial agents were developed, and these were called sulfonamides. They were actually developed from dyes. They observed that the dyes could be used and then as anti-infectious agents, and so they developed these sulfonamides from those dyes. These were the first truly antibacterial agents, and they were very, very effective against many different pathogens. But the problem with them was that they had many side effects. They were very toxic. And second of all, the bacteria that they acted against very rapidly developed resistance. After a while, the bacteria developed genetic mechanisms to evade the agent, to evade the sulfonamides. There was a spectacular period when a whole range of diseases in the 1930s were being effectively treated by sulfonamides. For instance, tuberculosis. It was the first time that tuberculosis could be treated with a chemical agent. They had very great success, but very soon resistant bacteria developed, on which the sulfonamides had no effect whatsoever. And then there was a drought in treatment for a very long time. Until actually the Second World War. Now, this is a long story. Okay? I mean, I'm not going to tell you a long story. I'm just telling you it was a long time for the development of the first true antibiotic. In 1928, this scientist, Alexander Fleming, was going away on holiday. Now, Alexander Fleming worked on Staphylococcus aureus, a very, very common bacterium, which is all over the place. We have it all over our skin. And under those circumstances, it causes no problem. But if Staph aureus ever enters into your body, it can cause huge problems. It's one of the agents that may cause puerperal sepsis, for example. But it's also the common agent that causes boils, but it can proceed to cause sepsis under various circumstances. So it was a widely recognized pathogen, and he was working, trying to find out ways in which to combat it. So he was going on holiday, and he left some plates of Staph aureus around his lab, and he actually, when he came back from holiday, this is what he noticed. You need to look really carefully to analyze what's happening here, but he recognized immediately. Here are the Staph aureus colonies in pure culture, which he had been working on. When he left, he probably had those colonies spread all over this plate here. While he was away, his plate got infected with fungus. And he immediately realized that if you look here, there's a boundary. There's a sort of circular boundary. Beyond that boundary, the Staph aureus colonies are normal size. Inside that boundary, as you approach the fungus, the colonies get smaller and smaller and smaller. And he hypothesized that what was happening was this fungus was producing a substance which combated the life of other organisms. So he dubbed it an antibiotic. He never did anything with it. Strangely enough, he never then made the leap. He published his results, however, and other people did notice. And by the Second World War, they had been realizing that they had a critical need for antibiotic agents to combat wounds, combat infection of wounds which people suffered during the war in battle. And in the 1940s, this was the first time that this substance, that this antibiotic that Fleming had discovered, was put into commercial production, into industrial production, not commercial, but industrial production. They could produce large amounts of it. Now, this fungus here is penicillium. That's its name, it's penicillium. And Fleming named this antibiotic substance penicillin. And penicillin production was first undertaken on an industrial scale during the war, and it had huge success. And it was very widely used, both then and after the war. It suffered, though, from the same problem that the sulfonamides did, but much more slowly. And that was that bacteria began to develop resistance to it. So after penicillin was made, they began looking at a wide variety of fungi and bacteria to see if they also produce these kind of substances. And they found them all over the place. Now, why would a fungus produce an antibiotic? For this very reason. Fungus competes with bacteria for nutrients and under normal circumstances. The penicillin normally grows in soil and all of this stuff. It competes with bacteria for nutrients in the environment and for space as well. So if it kills off bacteria in its environment, it has more space and more nutrient to grow. It's a competitive strategy by the penicillin, but the same strategy is used by many fungi and also by other bacteria. And this is where we get most of our antibiotics from. Often they are named for their source. So for example, streptomycin is extracted from organism called Streptomyces. And there's a whole wide range of different organisms which can produce these different substances. As a result, they began to produce a wide range of these, but they all suffered from the same problem, and that is that their overuse can lead to resistance. In addition, some of them are toxic, and this applies especially to the early antivirals. All of the agents I've talked about were actually antibacterial, but we did begin to develop substances which would combat viruses as well, but these have huge problems of toxicity. We'll discuss that when we talk about viruses. So the attempt to address these different problems has led to what is called the third golden age of microbiology. First golden age, Pasteur, Koch, all those people. Second golden age, development of the agents against bacteria. Third golden age is the attempt to develop new agents which lack the problems of toxicity and resistance. So let me just tell you, first of all, that the field of microbiology has exploded into different major subfields. Bacteriology is the study of bacteria themselves. Mycology is the study of fungi. Parasitology is the study of eukaryotic microbes. We will talk later about what prokaryotic and eukaryotic organisms are, and we will only very briefly, unfortunately, discuss the role of eukaryotic organisms. Virology is the study of viruses. I will discuss later when we talk about viruses how these were originally discovered. They were discovered very late in science. And we now enter the fourth golden age of microbiology, and that is the era of microbial genetics. All living things use DNA as their genetic material. And that the structure of DNA was first elucidated by these workers, Watson, Crick, Wilkins, and Franklin. She's always left off, but I'll explain when we discuss it what her role was. And the understanding of what the DNA structure is and how it functions has led to the development of a whole field of molecular biology. That is biology directed by the activity of DNA. And through that, we've been able to start to use methods of manipulating DNA, not only to further our understanding, but also to combat disease. And unfortunately, in this class, we will discuss DNA. We will not discuss molecular biology to a great extent. And unfortunately, we will also omit from this class the study of microbial ecology. Microbial ecology is the study of the role of bacteria in the environment, and in particular, the role of bacteria in recycling of elements and compounds so that they become available to biology in general. This is an essential role of bacteria and of microbes, because otherwise everything would get locked up in huge biological molecules, which would just last forever, and we would eventually be starved of the building blocks of life. Unfortunately, microbial ecology is omitted from our studies this semester. Okay, so with that, I'll finish for today, we begin our second block tomorrow, which is bacterial form and function. Okay, so please have a look through those slides. I'll post them this afternoon. Okay, thanks a lot.